this is our unknown presentation.
Throughout the semester, we have done multiple different tests in order to try and identify our unknown organism. This PowerPoint shows our step-by-step process on how we found out what our unknown organism is. And this is a picture.
The first test we did was the blood agar test, and that's where we tested to see which bacteria can partially or completely break down the blood cells and what bacteria cannot. So we tested these 4, and our unknown consumed the blood and created one big colony. And then this is our drawing and our notes of our results, and our unknown made like a massive colony and others didn't. And then here's the book's example of what it should have looked like.
Our second test was the mannitol salt agar. Mannitol salt agar differentiates between pathogenic and non-pathogenic staphylococci. It first tests for the presence of organism growth. It also tests for visible color change, and if the agar changes to yellow, then the organism is a mannitol fermenter and is disease-causing. Non-disease-causing staphylococci grow but don't change the color of the medium. And we tested these 4 organisms, and our unknown, it had like a small bit of growth, and so we tested it again because we didn't know if the growth was from this other organism or if it was from our unknown.
The next test that we did was the eosin methylene blue agar, and that is a medium that is selective and differential. It contains 2 dyes, the eosin and methylene blue. This makes a medium selective by inhibiting the growth of gram-positive bacteria, but allows many gram-negatives to grow. It also contains the sugar lactose, which differentiates between non-lactose fermenters, lactose fermenters, and heavy lactose fermenters. So we tested E. coli and Pseudomonas and our unknown, and our results are over here, and they kind of mixed a little bit, so it's hard to tell, but our unknown was a non-lactose fermenter.
The next test was the McConkey agar. The McConkey agar test is used for isolation and identification of gram-negative organisms. This media contains selective agents like salts and dyes, and lactose as a differential agent. And we tested these 4 organisms, and our unknown did not grow on this, and which meant that it was gram-positive. So this is our drawing of what it looked like.
The next test is a catalyst test, and this is the Oxygen and Growth Catalase Lab was testing to see if our unknown is a catalase of oxygen. If bubbles appear shortly after placing the hydrogen peroxide on the side, then it is positive. If no bubbles appeared, then it would be negative. We tested our unknown on this test, and we observed that our unknown did bubble after the hydrogen peroxide was placed on the side. Therefore, our unknown is a positive for catalase of oxygen.
The next test was the oxidase test, and it's trying to see if your bacteria is a positive or negative oxidase. If positive, the bacteria will turn bluish purple, and if negative, a blue ring will develop around the sample. And we only tested our unknown, and we observed that when the liquid was added to our unknown, it turned a blue color, and therefore our unknown has a positive oxidase reaction.
The next test was the Kirby-Bauer method, which is a test to see what antibiotic would react and treat our bacteria the best. With this test, we only tested our unknown as well. The procedure was to spread out the unknown all over the plate and place 8 discs of antibiotics around the plate evenly. We let our plate then sit for about 48 hours and then measured the distance of the ring where the antibiotic ate away our bacteria around each disc. We believe that the chlorophenol antibiotic will best treat our unknown.
This is the gelatin hydrolysis test. The process of this test involves transferring the chosen organism into a tube of medium that is a combination of both broth and gelatin. When inoculating, you have to make sure that you stab the lip all the way to the bottom of the tube, and the tubes are then inoculated for 24 to 48 hours. Once removed, they are placed in the refrigerator for 20 to 30 minutes. The gelatin hydrolysis test is testing for the presence of gelatinase. Gelatinase breaks down gelatin into the amino acid or peptide level. If the medium gels, the organism is gelatinase negative. If the medium remains liquid, the organism is gelatinase positive. So we tested these 3 organisms are unknown, and its results were liquid, which means that it does produce gelatinase.
Okay, for the starch hydrolysis test, we inoculated the starch agar plates with the organism of interest and then you would add the iodine, and if the test was positive, then there would be no color after the iodine was added. And we tested these 3 bacteria, and then for our unknown, it was mostly clear, so it means that it does hydrolyze starch.
Okay, for the next, we did casein hydrolysis. If an organism produces and secretes casease, then casein in the agar surrounding the colony will be hydrolyzed and a clear zone will surround the colony. If the organism does not produce casease, then the colony growth will appear on a white background. So we did these 3, and then for unknown, there was no change in the agar, so that means that the hydrolysis casein was negative.
The next test we did was phenol red, and the phenol red broth is used to determine an organism's ability to utilize a specific sugar. It includes phenol red, the sugar of interest, salts, and peptones. The tubes that contain the medium are Durham tubes, which trap any gases released during the fermentation. And so for this test, we tested all of these bacteria, and then for each bacteria, we tested for glucose and lactose. And then for our unknown, for the glucose, the color changed to yellow-orange, and there was no gas production, so that means it's early reversion. And then for the lactose, the color was red, like a red/clear color, and there's also no gas production, which means it's a controlled tube.
For the triple sugar iron agar, this test was used to study for the carbohydrate fermentation capabilities of an organism. This medium consists of glucose, sucrose, and lactose, along with a pH indicator, 2 sulfur sources, and an iron salt. When the acid is released from this fermentation, it'll cause the pH indicator to change from red to yellow. So we also did these 3, and then for our unknown, the slant color was yellow, but the butt was yellow with bubbles. So there was no H2S present, and then for the interpretation, we said glucose and lactose or sucrose fermentation gas produced.
So the next test was the litmus milk test, and the litmus milk test is used to identify a wide range of bacteria. It contains a pH indicator along with sugar and protein. This indicator can also show a range of reactions from changes in pH to changes in oxidation reduction status. So we tested these 4 bacteria, and then our unknown, which is also right there, it resulted in a medium purple color, and then we interpreted it as an alkaline reaction.
Okay, so for the last test, we did the intrapleury test. For this, there are 12 compartments in this intrapleury test that contain different mediums that run different tests. So how we did it was, you take the white cap off and you put your unknown organism on that, and you pull a needle through all 12 compartments and push it back through. And then you let it sit for about 48 hours, and after the husband, it is compared to an uninoculated tube and to determine which compartments are positive and which are negative. The results are used to generate a 5-digit code, and this code is used to determine the organism identity.
So our unknown is Protus vulgaris, and it's a rod-shaped gram-negative. It's resistant to antibiotics, has significantly increased over the years. It is a 3rd-generation cephalosporin. It is a facultative anaerobe, and it has the ability to degenerate urea to ammonia.
Okay, so for our conclusion, the transmission and infection, basically it can be spread through soil, water, and fecal matter. It can be transmitted directly or indirectly through a contaminated person or object, and it is known to cause urinary tract infections.
So the treatment for it is antibiotics, and these are some of the antibiotics that can treat it. And then you can prevent it by good hygiene and going to the bathroom regularly to flush out bad bacteria in the urinary tract.